So thank you very much, Chris. So we're going to get even a bit more basic epidemiology. Because when we're going from Africa to the other part of the world, there's a lot less known actually about the infectious reservoir. There's even very little known about gametophytes of non-falciparum species. In the wonderful review that Tern and Chris written a couple of years ago, there really is only a very, very small fraction of that review deals with Plasmodium vivax, and that illustrates how little we know about these essential sexual stages of the non-falciparum parasites. So basically, what we found, obviously when we want to eliminate, we need to know where transmission is. We need to understand that better, and that's really the basic rationale why we started that work. But at low endemicity, as it is found in most non-African parts, we've also found out over the last couple of years that we have a very large pool of asymptomatic infections, even at very low transmission densities. And we also know that transmission becomes very focal, both in space, but possibly also in specific groups of people, because they have different exposure due to behavioral characteristics. For instance, in Southeast Asia, the main risk group are adult males, because due to occupational hazards, they are the ones that most often go to where mosquitoes are present. And so there is very little data from these settings on which people do actually constitute the transmission reservoir. And there is almost no data on how much these very abundant asymptomatic infections in these areas may contribute to maintaining transmissions. So we set up a study, which we call the TransEpi study, for transmission epidemiology study, whose overall goal really was to understand the dynamics of transmission in 3 different settings. And these settings are the Southwest Pacific, Thailand, and Brazil. And we set out to do 3 different types of study. The first study was very simple, just to see cross-sectionally in the population where are gametocyte carriers and what's the relationship between people carrying parasites and gametocytes. We then set up a number of longitudinal cohort studies to look at the temporal dynamics of both types of infections, asexual and sexual infections. And lastly, we also did, on a much smaller scale and not as sophisticated as was just shown by Tern, to do infectivity studies to try to find out what is the infectiousness of these parasites. And we've also been working on improving some of the detection methods, and Ingrid Felger will talk more about that, and Matt Marty is also talking more a bit about these aspects. So where we're working really, and the people that are part of this consortium, it's coordinated from Barcelona, and we have lab groups in Switzerland, in Basel, at the Walter and Eliza Hall Institute, and we're collaborating with the group at Harvard. And we have our field sites in Manaus, at the Fundação de Medicina Tropical, in Mahidol University, and we're working with the PNG Institute of Medical Research in PNG. And we have a field site that's run by the Walter and Eliza Hall Institute in the Solomon Islands. So the first studies is we've done a large number of cross-sectional surveys over the last 3 years. And in total, we have included 16,500 people into these cross-sectional surveys. This is ongoing work, and I will only present data from Thailand and from 1 of 2 surveys in Brazil and the first survey in Papua New Guinea. What we've done with all these samples in a standardized methodology that was set up at each field site in quality control with Basel as the reference laboratory. So we're collecting DNA, we're collecting blood, extracting DNA. We're then using a gatekeeper 18S PCR that amplifies genus-specific products because we have relatively low prevalence and this is more cost efficient. We then go into the ones that are positive by genus. We then do a PF and PV qPCR specifically for PF and PV. These samples are getting genotyped for other assessments, but importantly then all the PCR positive samples are getting extracted for RNA, and then we're testing for gametocytes for RNA. So differently from what Toyn has shown, we are not doing a direct RNA detection assays in all samples, including the ones that are PCR negative. And that's for a simple cost reason. If you have a prevalence of 3-4%, then it becomes very, very expensive to do an RNA extraction on 95% negative samples. So this is the 3 surveys that I'm currently talking about. So when we're looking at the prevalence by PCR, we're seeing that in Thailand and Brazil, Vivax is clearly the predominant species, but we really have only 3 to 4% prevalence, while falciparum is found in about 1.5% in Thailand and basically about 0.8% in Brazil. In PNG, on the other hand, it's still a moderately endemic setting where we have between 10 and 15, 16% prevalence for PF and PV and slightly more PF. When we're looking at in terms of microscopy wasn't done in Thailand, but in the other two sites. So in both sites for PF and PV, roughly have half the parasites that are only detectable by PCR in terms of Vivax, and in PNG also for falciparum. However, in Brazil, every single PF infection that we found, which were very few, was also positive by microscopy. We had 0% submicroscopic falciparum infections in the Brazilian setting. When we looked at how many of these samples were asymptomatic, we find for both species that between 80 and 90% of infections that we detected did not report any form of febrile illness. They were neither feverish at the time of the survey, nor did they report that they have been feeling feverish in the previous 48 hours. So when we then looked for gametocytes in the positive samples, what we're seeing is this pattern that we have basically with around 50 to 80% of our samples come up positive for gametocytes. Slightly more for falciparum. In particular, in Brazil, we have almost all samples, because they are microscopy positive. I think all but one, there were 11 people in the whole survey of over 2,000 people with falciparum, and 10 of those are gametocyte positive, and only one was not positive by gametocytes. But also in PNG, we have slightly higher prevalence of falciparum gametocytes than Vivax gametocytes. Why is that? The reason comes from here, and it's related to the parasite densities. So what you're seeing on, first we go walking through the PNG data here. Let me get them. Here we go. So this is the number of 18S copies, which corresponds to the total parasitemia in the blood. And this is the probability to be positive by RT-qPCR for gametocytes. So you see that there's a very strong relationship that as you get more overall parasitemia, your probability to detect gametocytes by qPCR raises dramatically. What you also notice is that the distribution of densities of Vivax is much more on the lower side compared to falciparum, and that when you're looking at the same densities, we actually have a higher proportion of gametocytes in Vivax infections compared to falciparum if we match them for densities. And you see these not very well in Thailand because this is too few cases here for getting a good fit for falciparum, but we have a very similar curve between Vivax for both Thailand and Brazil. When we then look in the positive samples, the ones that are positive for gametocytes, what is the prevalence of infections, of densities of gametocytes, we find very tight correlations in Vivax of how many PFS25 transcripts we find per 18S copy. When we look at the ratio and plot this is almost a Gaussian distribution. So it looks like there is a constant proportion of parasites in the bloodstream of Vivax that are gametocytes. Irrespective of the density you have, you always have roughly the same gametocytes per total parasite biomass. There is very poor correlations of the same for falciparum. That it does not map very well your total gametocytes versus your total parasite biomass. We then tried to figure out exactly to quantify this, and we've done trendline analysis where we took samples with known numbers of gametocytes by microscopies and did dilution series. And we have done one that was published in a paper by Wampler et al. With Ingrid's group, where we used cultured falciparum parasites. We did dilution series, related the 18S gametocytes by microscopy with the PFS25 copies, and we find this non-log-log linear relationship. And roughly on average, but that depends on the actual density because it's not a linear relationship. But if you're looking across most field samples, where most of the samples are, you have roughly 80 copies of PFS 25 per gametophytes. It ranges from maybe 50 to 300 depending on what your parasitemia is. So we have a non-linear relationship. When we're doing the same thing for Vivax field samples that were prepared in Thailand and then diluted, and we had different methods of conserving the RNA, ones in trisol and ones we did the samples into RNA protect, we find a much, much lower number of transcripts per gametophytes, only about 7 to maybe 10 transcripts per gametophytes. We do not know at the moment whether that is because the parasite has an overall much lower expression, or whether the RNA in Vivax gametocyte is not as stable in terms of what we're doing for the extraction protocol, where we're having DNA treatment protocols where we do lose a certain amount of RNA, and therefore we may actually be selectively losing transcripts more in Vivax than not. But this has very important consequences when we are comparing Vivax and falciparum gametocytes. So the conclusion of these initial studies and data we have is that we roughly have between 50 and 70% Vivax samples and 60 to 90% of falciparum samples are positive for gametocytes. For Vivax, it's very closely related to parasitemia, but not for falciparum. But the comparison between the two species is hampered by diagnostic uncertainty. Vivax gametocytes are much more difficult to detect because Vivax infections tend to be of much lower parasitemia. We seem to have lower expression or lower stability of the PAVS25 transcripts, which again reduces the absolute limit of detection that we have with our PCR assays. So these estimates here are an underestimate of the true gametocyte number compared to what we see in falciparum. And that's nicely illustrated by a study that was done by Marcelo Urbano Ferreira in Brazil, where he took larger volumes of Vivax blood and did it directly into liquid nitrogen. And then if you extract RNA from that, you have 100% gametocytemia in Vivax samples, irrespective of parasite density. So a lot of what we're finding here, the difference is probably our inability to detect very low levels of gametocytemia. So these technical shortcomings are essential when we are trying to interpret the data from these kinds of studies, and also the difference between the two species, because it's not the same characteristics. There were two posters presented here earlier today that gave more details on two of these cross-sectional surveys, one by Christian Köpfli and one by Andrea Kuhn. So the next part is we then started to do the infectivity studies on mosquitoes and ask, quite similar to Toyn, the main question really was, are these asymptomatic low-density infections infectious to mosquitoes? We did that by doing PCR-based mass surveys to detect asymptomatic carriers. That is a huge amount of work. If you're working in Thailand or Brazil with 3% prevalence, you have to bleed 100 people to do 3 feeds. So if you're doing 100 feeds, you have to bleed 3,000 people before you have 100 positive blood slides. So that's why accruing that data is extremely laborious. So we're doing these surveys, we go out, take a finger prick, we run them on a PCR on a lamp. Everybody that comes positive with PCR lamp, we go back 48 hours later, we reconsent them, we then take a 5 ml venous blood samples, we keep it warm at 37 degrees, rush it back to the lab, and then perform a standard membrane feeding assay. And these studies are ongoing in Thailand, Brazil, and have just started in PNG, and I'll be presenting a bit of data from Thailand and Brazil. So in Brazil, there was a poster also here at the conference, a late-breaking poster by Kaylin Campos and Welton Monteiro from Brazil, who presented their data that we have so far accrued on 35 symptomatic and 42 asymptomatic Vivax samples. So what we found is that 85% of symptomatic Vivax samples infect mosquitoes. So symptomatic Vivax samples are extremely infectious to mosquitoes. You have very few that do not infect mosquitoes, and you have high numbers of oocyst numbers. We also found that 19% of the asymptomatic infections did infect mosquitoes, including 2 samples that were not positive, where we had no gametocytes, but where we had no the 3 samples had no gametocytes by microscopy, and 1 that was actually PCR, that was positive only for the RT-PCR. The infection and oocyst rates seem to be correlated with gametocyte density, at least up to a certain level, and then it becomes more or less constant. But there is not enough data yet to really draw good inferences on these relationships, so we need to collect more data. This is the current state of what we've been doing, what Chetsimon Sattabongkok's group and her team have been doing in Thailand to date. And we've had done 50 symptomatic cases and 42 asymptomatic cases, and we find that 78% of symptomatic cases do infect. Very interestingly, in Thailand, there's a huge difference in falciparum to vivax. Basically, all symptomatic vivax patients infect mosquitoes, but only 2 out of 12 symptomatic falciparum patients infected mosquitoes. And that's because of the very highly efficacious Thai health systems, where most patients do show up for treatment before they have mature gametocytes circulating in the bloodstream. When we look at asymptomatic infections, we find overall about 10% of asymptomatic samples infect mosquitoes, but there is a big difference, and we found a lot of mixtures of species, including PM and P. nosai among these asymptomatic infections, and there is not enough data yet to really draw any firm conclusions between different species. But it shows that there is somewhere between 10 and 15% or maybe 20% of asymptomatic carriers can actually infect mosquitoes if fed in a standard membrane feeding assay. So from the infected studies, we find that these infections do actually infect, as Toyn has shown for the African side, as well for falciparum, and that there seems to be this relationship for vivax, at least, with gametocyte density and infectivity. There is, however, a bit of a question that came up to the way we worked. We are not, similarly, I think, with Toyn. The membrane feeding assays are a good tool. They make work a lot easier. They are reproducible. But there is a question whether we really should be doing skin feeds to get an accurate estimate of what infectivity is, and we would like to look at that question also in a non-African setting and do direct feeds on infected patients. So the next step of what is going on at the moment is we have completed all these longitudinal studies, and we are analyzing now, looking at the dynamics of parasitemia versus gammaseptemia over an entire year. Again, these are large numbers. These are 3,700 people that we follow for a year, because again, there's a small percentage that actually do get infected, so that we need to get enough data. We also are conducting spatial risk analysis, both of infection and potentially infectious individuals, to try to kind of see whether within our study sites, whether transmission is concentrated either in specific locations or in specific populations, whether that's males or that is adolescents or other groups of people. And we will continue infectivity studies and hopefully have a larger, better data set to draw more strong conclusions. So overall, the overall conclusion then really is that I would say that all vivax gametocytes have gametocytes, all vivax infections. We cannot always detect them, technically. But if we had large enough volumes of blood and we can conserve the RNA good enough, we would find that every vivax infection has gametocytes. In falciparum, in our study, the gametocytemia is much less closely related to parasitemia than for vivax. And we have shown now that asymptomatic vivax infections are infectious and that they do constitute a potentially important part of the transmission reservoir, considering that they make up 90% of all vivax infections. I'd like to acknowledge our collaborators that have directly contributed to this work, in the Walton Lights Hall Institute, the PNG IMR, the Foundation of Tropical Medicine in Manaus, Mahidol University, the Swiss Tropical and Public Health Institute, and at the Barcelona Center for National Health Research. And I would like to also acknowledge our colleagues in Harvard, with whom we are working on for further genetic studies on some of the samples that we have collected. Thank you very much.
Thanks, Ivo. We'll take a couple of questions from the floor before we move on. Jeff?
Ivo, point of clarification. With your Vivax symptomatics, which were transmitting very well, when were they actually tested? Because Richard Carter and Carmine Mendes had shown a long time ago that there were stages where the gametocytes were relatively non-infective. It varied over a relatively short period of time.
The majority of the samples we get have a lot of trophs. And that has made it incredibly difficult for us to try to get pure Vivax gametocytes for transcriptional studies. So, and that's probably because people have a paroxysm, they feel very ill, and 12 hours, 18 hours later, they show up for treatment. So a large part of our patients would probably be within about 12 to 24 hours of having had the paroxysm. And that's when the samples get collected, and that's the samples that are being fed.
Nick White, Mahidol, Oxford Research Unit. Ivo, could you comment on oocyst numbers in the asymptomatics versus the symptomatics?
Substantially lower. Substantially lower in the asymptomatics than to the symptomatics.
So you could use that, compare that to wild-caught oocyst numbers and derive or approximate how many?
Most of the positive mosquito, we have had some samples in Brazil that had surprisingly high numbers of oocysts, where we had 10, 15, or even 20 oocysts in a mosquito, which really got us very confused why we would find so high oocyst numbers when the gametocyte numbers seem to be low. But in most samples, the numbers are low, 2, 3 oocysts per mosquito.
Ivo, it was just a couple of points. I'm not happy with the use of the word asymptomatic when you mean submicroscopic, because you can be asymptomatic and have a very high load of parasites. So it's maybe just nice to use the word submicroscopic for what you question you're asking, is how many people with submicroscopic malaria are transmissible. This is what you really want to ask, isn't it?
I agree with you, and we actually haven't. I maybe been a bit fussy with my speaking, but what you saw in the slide, where we roughly have 50% submicroscopics, and we have about 80% asymptomatics. The two groups are not fully overlapping.
Can I make a comment just in defense of our ex-head of the department, Frank Hawkins, who had this idea that the infectivity varies? I don't think he meant it to be that it varies in numbers of gametocytes that suddenly disappear or sequester. P. falciparum gametocytes do sequester, so it's not surprising. It's their infectivity, it wasn't their number. It's they change infectivity from morning to evening. And that might be to do with the way we might be different between morning and evening, but without necessarily being sequestered. Thanks.
Okay, thank you. Thanks.